Welcome back to the Learning Studio. This is the Biological Sciences series, and today we explore three interconnected breakthroughs that changed modern science forever: PCR, DNA sequencing, and molecular diagnostics. If you missed our last lecture on recombinant DNA technology and cloning, the full playlist is linked in the description below. Here's a thought: how did we test millions of people for COVID-19 within hours, or decode the 3 billion letters of the human genome? The answer lies in PCR, sequencing, and molecular diagnostics. PCR, the Xerox machine of biology. Polymerase chain reaction, or PCR, was invented by Kary Mullis in 1983. He described it as DNA photocopying, a way to make millions of copies of a specific DNA sequence in just hours. PCR works in cycles. Denaturation, DNA strands are separated by heat. Annealing, primers attached to target sequences. Extension, DNA polymerase builds new strands. Repeating this cycle 20 to 40 times produces billions of DNA copies from just one template. Applications: diagnosing infections, COVID-19 PCR tests; forensics, matching DNA at crime scenes; research, cloning, gene studies. Without PCR, modern genetics and diagnostics would not exist. DNA sequencing, reading the book of life. While PCR copies DNA, sequencing tells us the exact order of nucleotides, A, T, G, and C. Sanger sequencing, 1977, was the first method used in the Human Genome Project. Next-generation sequencing, NGS, now allows entire genomes to be read in days instead of years. Case study, Human Genome Project 2003. Took over a decade and nearly $3 billion to map all human genes. Today, thanks to sequencing advances, a person's genome can be sequenced in a day for under $1,000. Applications include identifying mutations in cancer, studying microbial communities in the environment, tracking virus variants during pandemics. Molecular diagnostics, from the lab to the clinic. Molecular diagnostics applies PCR and sequencing directly to human health. Examples: infectious diseases, detecting HIV, tuberculosis, or COVID-19 with PCR. Genetic testing, screening newborns for inherited disorders. Oncology, finding specific mutations to guide personalized cancer treatments. Prenatal diagnostics, sequencing fetal DNA from maternal blood samples. Molecular diagnostics is the bridge between molecular biology and patient care, making medicine faster, more accurate, and increasingly personalized. Why this matters in biologicals: PCR, sequencing, and diagnostics are the microscope of the 21st century. Instead of looking at cells, we look at genes. They show us how life works at the molecular level and help us fight diseases before they spread. Past, present, and future relevance. Past: the invention of PCR and Sanger sequencing transformed research. Present: COVID-19 testing and personalized medicine rely on these tools daily. Future: portable sequencers, AI-powered diagnostics, and point-of-care PCR may revolutionize global healthcare access. So that's our journey into PCR, sequencing, and molecular diagnostics, the technologies that gave us the power to copy, read, and apply the code of life. In our next lecture, we'll shift from molecules to cells, exploring the future of medicine with stem cells and regenerative medicine. Welcome again to the Learning Studio. Don't forget to check the playlist link below and subscribe so you won't miss our next episode. Because once you understand these tools, you realize we are not just studying biology, we are rewriting it.